Hi, good morning, students. This is Avantika here. Today I am going to teach a topic of Introduction to Microbiology. So, let us first start with the topic of microbiology, that is, Introduction to Microbiology. Okay, let us first know what is the definition of microbiology. The definition of microbiology is it is a discipline of science which deals with the microscopic or submicroscopic forms of life. They are usually invisible to the naked eye, and microbiology is actually a word which has came from a Greek word. Micros means small, bios means life, and logos means small again. So this is the discipline of science which deals with the very microscopic or very microscopic forms, and they are usually invisible to the naked eye. They can be only visible to the naked eye with the help of a microscope. Without microscope, we are not able to observe these kind of microscopic forms. And these microscopic forms are basically bacteria, fungi, virus, etc. Another important thing is microorganisms were the first living inhabitants on the earth. So it was discovered by the scientists that the microorganisms were the first living inhabitants on the earth. They were found to be the first living inhabitants on the earth. Okay, now let us see what are the landmarks in the history of microbiology. What are the basic landmarks which were introduced during the 19th century? Firstly, it was the discovery of microscope. The first important landmark in the history of microbiology was discovery of microscope. So the discovery of microscope was studied by different scientists. It was first Roger Bacon in 1267 who developed a lens for the first time. So this was the scientist who developed a lens for the very first time. Then another two scientists, Janssen and Janssen, produced a crude type of microscope by placing two lenses together. So basically they created a crude type of microscope. They just placed the two lenses together in 1590. Okay, then came the Galilei in the 1610, who prepared a microscope with a focusing device called occhiale. The name microscope was first proposed by Faber in 1625. But the most important contribution which was done by Robert Hooke from 1635 to 1703, he made and used a compound microscope in the 1660s. He described his fascinating explorations in his classic Micrographia. So Robert Hooke was the first person who discovered the compound microscope, and it is very important in the history of the microbiology. So he explored his he described his fascinating explorations in his classic, that is Micrographia. Okay, now we can see this is the first microscope which was developed by Robert Hooke. Microscope used by the Robert Hooke. Now we can see that it is simply a eyepiece. Then barrel is here. There is a focusing screw, objective. Then there is a specimen holder to hold the specimens like slides, etc., from where we can observe by which we can observe the microorganisms, example bacteria. This is the water flask and oil lamp. So this is the place where specimen holder is a place where we can hold the specimen, and with the help of the eyepiece we can look into the compound microscope and we can observe the particular specimen. So this was the first microscope which was developed by Robert Hooke. Okay, now what were the other things which developed during this time? So the first discovery was of microbial life. A scientist named Antony van Leeuwenhoek in 1677 first recorded observations of microorganisms. So Antony firstly started studying the microorganisms. He described such tiny creatures as Dirkens or animalcula. So he was the first person to observe the microorganisms, and he named these tiny creatures as animalcula. Then came another scientist, O.F. Muller. He gave the first classification of bacterial microbes in 1773 and 1788 and coined the terms Vibrio and Monas for the certain forms. So he introduced the words Vibrio and Monas for the certain forms. Okay, now there came the spontaneous generation or abiogenesis. This was a theory which was introduced by the few scientists early, because the scientists of the ancient times were not knowing anything about the microorganisms. They were just exploring the field. So, firstly, they believed that all living matters could spontaneously generate from the non living matter. This belief was referred to as the doctrine of spontaneous generation or abiogenesis. So the scientists in the early times, they thought that all the living microorganisms can get generated from the non-living matter, and this is known as abiogenesis. The idea of spontaneous generation continued until the mid 19th century with great oppositions against it. So this idea was opposed by various scientists and many people, but this idea continued till the mid of the 19th century. Okay, now how does this end of the debate happen? Actually, the Louis Pasteur ended this debate. Louis Pasteur in 1861 conducted a series of experiments to prove that there is no spontaneous generation of microbes. So Louis Pasteur was the person who actually ended this debate, and he introduced that there is no such doctrine, that is spontaneous generation of microbes. So how he performed few experiments. On the basis of that, he proved that there is no such abiogenesis process. So what was the experiment done by the Louis Pasteur? The experiment which was done by the Louis Pasteur was a swan neck experiment. So during this process, he took various types of broth like yeast water, sugared yeast water, urine, sugar beet juice, etc. in long naked flasks, and then softened the neck of the flask under a flame and drew it out in the shape of S looking like the neck of a swan. So firstly, he took the various types of broth such as yeast water, and he then softened the neck of the flask under a flame and he made it like a S shape. So he softened the neck of the flask under the flame, and he made it like the S shape. He made it in the form of the S shape, looking like the neck of a swan. So the swan neck experiment was performed by was performed like this. The broths of these flasks were boiled until they steamed through the neck. So these broths were taken in these flasks and they were boiled until the steam was coming out of the necks, and then these broths were cooled. After cooling, the broths so treated in the flasks did not decay, and there were no signs of microorganisms in them after days, weeks, and even months, though they were open to free air. So after Louis Pasteur boiled these flasks and then he cooled these broths, he came to noticed that there are no decay or there are no signs of microorganisms in the broth even after days, weeks, and months, even if they were exposed to the free air. So he was the first person to conclude that with the help of this particular swan neck experiment, he observed and told that swan necks of the flask trapped airborne microorganisms before they could reach the broth and flourish in it. So because of this kind of neck, the swan neck, the airborne microorganisms were trapped into these neck, and they were not able to reach the broth and were not able to continue their growth. So the broths in these flasks were open to free air but free of microbes for very longer periods. Therefore, he discredited the doctrine of spontaneous generation. So these broths were open to free air, but the microbes, as I have already told, that these microorganisms were trapped in the neck of the flask, which was S-shaped, and so he concluded that there is no such doctrine of spontaneous generation. So he just ended the debate. Okay, now let us know about the Louis Pasteur mode. Louis Pasteur from 1822 to 1895, he is considered as the founder of microbiology. He also discovered the process of fermentation. So Louis Pasteur was founder of microbiology, and he concluded the abiogenesis process. Another process which he discovered was fermentation. Now let us know what is fermentation. Fermentation is a biological process that breaks down carbohydrates into alcohols and organic acids. So here it breaks down the carbohydrates into alcohols and organic acids with the help of a specific type of microbes. So this fermentation actually occurs by the help of the specific microorganisms. He also introduced aerobic and anaerobic bacteria. So there were different kind of bacteria. For every specific kind of fermentation, these specific kind of bacteria were introduced for the fermentation. He also described the process of pasteurization. Now let us know what is pasteurization. During the fermentation processes, for desired products, certain undesired microbes grew in the ferments. So, Louis Pasteur noticed that during the fermentation process, for the desired products, the fermentation process created large amount of the desired products. But during this process, certain undesired microorganisms were introduced into the fermentation broth, were growing in the fermentation broth. So to avoid this, Louis Pasteur suggested that heating could be used to kill the undesirable type of microbes growing in ferments. This came to be recognized as pasteurization. So he suggested that if we are going to heat the complete broth or the during the fermentation process, it will kill the undesirable type of microbes which is growing in the ferments. So this particular process came to recognized as pasteurization. For example, we can say during the production of wine vats, rod-shaped bacteria grew in wine vats and produced lactic acid that caused souring of the wine. So, like rod-shaped bacteria was unwanted and it caused the souring of wine. So, it was reduced by the help of by introducing the heating. By the heating process, the undesirable microorganisms were killed. Today also, heating at moderate temperature to kill high percentage of microbial population was known as the pasteurization, and pasteurization is widely used in dairy industries. Okay, now comes the germ theory of disease. During this time, germ theory of disease was also introduced and studied by different scientists. So the first satisfactory demonstration of the probable causal relationship of organisms to diseases was given by Benedict Prevost in 1807. So he was the first scientist to study the relationship of the microorganisms to diseases. Secondly came the C.J. Davian, showed that bacteria are invariably present in diseased animals but are undetectable in healthy ones. So he studied, and he said that bacteria are actually present in the diseased animals as well as in the healthy ones, but in the healthy ones they are undetectable, and the diseases can be transmitted to healthy animals by inoculation with blood containing these bacteria, but he could not prove that. So he said that if we are going to inoculate these healthy animals with this contaminated blood, they are going to get the disease and the microorganisms can be detected by this. So, but he was not able to prove the full concept. Then came the Robert Koch in 1867, who solved the problem and confirmed the germ theory of the disease. The germ theory of the disease is also known as the pathogenicity test or the Koch's postulates. So let us know what are the Koch postulates. So the first point is in the Koch's postulates are the organism must be associated with the disease in the in all the disease hosts examined. So firstly, the organism which is causing the disease, it should be associated with the disease in all the disease hosts examined. That means ये जिस किसी में भी ये डिज़ीज़ कॉज कर रहा है उसमें ये एसोसिएटेड होना चाहिए और उसमें वो दिखना चाहिए. The organism must be successfully isolated and grown in pure culture on nutrient media. Its growth characteristics be recorded. So in his second point, he told that organism can be successfully isolated. The organism which is actually causing the disease, it should be isolated and it should be grown in a pure culture media, on a nutrient media, and its growth characteristics should be recorded. Now the third point, the organism should be isolated from pure culture and must be inoculated on the healthy host on which the disease appears. So in third point he says that this particular organism which is actually causing the disease, it should be isolated from the pure culture and it must be inoculated on the healthy host on which the disease appears. So the particular organism should be able to show the disease and it must cause the same disease on the inoculated healthy host. Now if we are growing it into the pure culture, it must be taken from there and it should be inoculated into the healthy host in which it should be able to show the disease. Now the fourth point, he said that the organism must be isolated from experimentally disease host during the step 3 and grown in pure culture again. So the organism which is infected by this disease or we have inoculated this disease, from the same organism it is isolated and again it is grown in the pure culture. During this pure culture, the organism should be able to show the same characteristics of its growth which was shown during the second step. So here also, after inoculating from the diseased animal and growing in pure culture, the organism should show all its characteristics and also the symptoms of the disease of the particular disease. So this was the pathogenicity test or the Koch postulates. Now other microbial techniques were also introduced during this time. So let us know what is pure culture concept and other microbial techniques. Firstly, we should know what is a pure culture. A pure culture is one that contains only a single kind of microbial population grown from a single cell. प्योर कल्चर एक ऐसा कल्चर होता है जो कि सिंगल काइंड ऑफ माइक्रोबियल पॉपुलेशन से ग्रो करता है एंड इट इज़ नॉट कंटैमिनेटेड बाय एनी अदर बैक्टीरिया. इट इज़ प्योरली a single kind of microbial population. So who introduced it? Firstly, it was Joseph Lister in 1878 who first obtained pure culture of bacteria using cereal dilutions in liquid media. Joseph Lister was the first scientist who discovered the pure culture of bacteria with the help of the technique called as cereal dilutions. He took a fluid containing a mixture of bacteria, diluted it with sterile medium, and delivered it into a container of sterile milk by syringe. So during his experiment, what did he do? He took a fluid containing a mixture of bacteria and diluted it with sterile medium. He took a mixture of bacteria, different kinds of bacteria, into a flask, and then he diluted it with sterile medium. He performed the cereal dilutions, and after that he delivered it into a container of sterile milk by the help of the syringe. Now he kept it for incubation. After incubation, he found that there was single kind of bacteria growing in container identical to their parent cell. So after incubation, there was only a single kind of bacteria. No other bacteria was found in that particular container. Only a single kind of bacteria was found in that container identical to the parent cell. So it was this is known as pure culture concept. Okay, now during this time there was also many kind of explorations in the in the field of medical microbiology. So what were the main important contributions in the field of medical microbiology? Let us see. The first is the causation of diseases. So during this time, reports regarding causative agents of animal and human diseases started pouring in during the second quarter of the 19th century. सो इन 19th सेंचुरी में ये देखा गया कि ऐसे क्या एजेंट्स हैं जो एनिमल और ह्यूमन डिजीज़ को कॉज कर रहे हैं, और उनकी स्टडी शुरू हुई. इन द सेकेंड हाफ ऑफ द 19th सेंचुरी, ड्यू टू अवेलेबिलिटी ऑफ बेटर माइक्रोस्कोप्स, Pasteur and his workers turned towards this area of microbiology, which led to several important discoveries. So in the second half of the 19th century, जब better microscopes आ गए, तो Pasteur and his workers इस field में, that is in the field of medical microbiology, he did a lot of discoveries in the field of medical microbiology. The first important definition which he introduced was immunization. Immunization is the artificial, what is immunization actually? It is the first artificial induction of immunity to a disease. Okay, another important thing which was discovered during this time was surgical antisepsis. In this also, the Joseph Lister, a British surgeon, reasoned that the post-surgery infections might well result due to the microorganisms present in air that enter the tissues exposed during operation, and revolutionized surgical practices in 1867 by introducing antiseptic principles. सो जोसेफ लिस्टर, जो कि एक ब्रिटिश साइंटिस्ट था, उसने सबसे पहले ये देखा कि आफ्टर द सर्जरी बहुत सारे इन्फेक्शंस होते थे टू द पेशेंट बिकॉज ऑफ द सर्टेन माइक्रो ऑर्गेनिजम्स, क्योंकि टिश्यूज़ एक्सपोज हो जाते थे ड्यूरिंग द ऑपरेशन. सो उसने सर्जिकल प्रैक्टिसेस को रेवोल्यूशनाइज किया और बहुत सारी ऐसी एंटीसेप्टिक प्रिंसिपल्स को स्टार्ट किया जो कि आज भी यूज़ किए जा सकते हैं. He used carbolic acid as a disinfectant and adopted several other antiseptic procedures which are still in practice today. So carbolic acid उसने यूज़ किया जो कि डिसइंफेक्टेंट करने के लिए सर्जरी इक्विपमेंट्स को, ऑपरेशन थिएटर्स को डिसइंफेक्ट करने के लिए कार्बोलिक एसिड का यूज़ किया गया, और अदर एंटीसेप्टिक प्रोसीजर्स वर ऑल्सो यूज्ड ड्यूरिंग दिस टाइम. Okay, another important discovery which took place in the field of microbiology was a rise of chemotherapy. Chemotherapy was also introduced during this time by a scientist, German physicist, who is known as Paul Ehrlich. He undertook extensive studies to search synthetic chemicals having curative properties for pathogenic microorganisms. सो पॉल ने सबसे पहले देखा कि पैथोजेनिक माइक्रो ऑर्गेनिजम्स को क्योर कैसे किया जाए जो कि बहुत सारे डिजीज़ को कॉज कर सकता है. सो ही स्टडीड द क्यूरेटिव प्रॉपर्टीज़. ही ट्राइड टू क्योर द डिजीज़ कॉज्ड बाय डिफरेंट पैथोजेनिक माइक्रो ऑर्गेनिजम्स, एंड दस ही इंट्रोड्यूस्ड द टर्म कीमोथेरेपी टू कंट्रोल वेरियस इन्फेक्शियस डिजीज़. During this time, the plant pathology was also introduced. During the early decades of 19th century, specific fungi was found which can cause the disease of wheat and rice. So in plants also, the specific bacteria were found, they were studied, and also plants were studied, and the relationship of both the bacteria and the plant was studied, that which particular bacteria is causing which particular type of disease. So another scientist, Burrell, in 1878 for the first time showed that the fire blight disease of pears was caused by a bacterium known as Erwinia amylovora. So he discovered that the first fire blight disease of pears में जो fire blight disease होता है वो एक specific bacteria से cause होता है, and the name of the bacteria is Erwinia amylovora. It was a phytopathogenic bacteria. Okay, another important discovery which occurred during this time was the discovery of viruses. So discovery of viruses was also studied by different scientists. The firstly, it was studied in tobacco crop. It was found that tobacco crop in Holland was struck by a severe disease around 1870. Adolf Meyer began his studies on this disease about 1880 and published his results in 1886. So he was the first person to study the discovery of to study the viruses, and he was also the first person to introduce a new discipline which was later recognized as virology. So he introduced the term virology. Okay, lastly, there was also explorations in soil microbiology. The fields of soil microbiology was explored during the very last part of the 19th century. In soil microbiology also, a very large amount of explorations was done. The establishment of the principal roles that microorganisms play in the biologically important cycles of matter. So, the various studies were done regarding the microorganisms which play a biologically important role in the various cycles of matter, such as nitrogen, sulfur, and carbon. So माइक्रो ऑर्गेनिजम्स और बायोलॉजिकली जो बायोलॉजिकल साइकिल होते हैं उनके बीच जो रिलेशनशिप होता है, इट इज़ कॉज बाय वेरियस माइक्रो ऑर्गेनिजम्स. सो ये रिलेशनशिप स्टडी किया गया by the help of two important scientists, that is S. Vinogradski and M.W. Bejring. The greatest contribution of Bejring was a new technique called enrichment culture technique. This technique was to isolate and study various physiological types of various microorganisms from natural samples and through the use of specific culture media and incubation conditions. So this particular scientist, he introduced a technique which is known as the enrichment culture technique. इस culture technique में पर्टिकुलर बैक्टीरिया को आइसोलेट किया जाता है, उनके वेरियस फिजियोलॉजिकल टाइप्स को स्टडी किया जाता है, और वेरियस माइक्रो ऑर्गेनिजम्स फ्रॉम नेचुरल सैम्पल्स थ्रू द यूज ऑफ स्पेसिफिक कल्चर मीडिया, natural samples such as soil. So the particular bacteria in soil was which was causing the disease or something like that. The microorganisms from these natural samples were isolated by the use, and they were grown in specific culture media and incubation conditions. And with the help of this scientist Bejring was able to introduce was able to isolate these various microorganisms in soil. So he greatly contributed in this field, that is soil microbiology. So students, this is all for today. In our next part of video, we will discuss the scope of microbiology and the careers related to microbiology. Thank you very much.